Assalamu alaikum. So we are going to talk about now the mechanistic function of Polycomb group and trithorax group. Okay. So what we just covered, that it was actually discovery by Thomas Genoan's lab that SUR3-9 is a histone H3 lysine 9 methyltransferase, which means, so they proved that, the lysine 9 in histone H3 N-terminal tail, it gets methylated. This is the lysine 9 methyl mark by SUR3-9, and that was due to the set domain, the specific amino acid sequences within the SUR3-9, which we talked about that it's also conserved in enhancer of zest and trithorax protein. It was, through sequence homology in 90s they discovered this. Now, they also showed that this lysine 9 methyl mark is recognized by the chromodomain containing protein HP1, which makes perfect sense because both SUR3-9, HP1, they were discovered in position effect variegation or suppressor variegation screens in fruit fly. And that's how they together constitute the, or formulate or contribute to the constitutive heterochromatin. Now, since the set domain was also found and it was known that it's present in enhancer of zest as well as in trithorax, so this discovery by Thomas Genoan's lab, it sparked a race to discover if other set domain proteins also are histone methyltransferases, because nothing was known except binding of Polycomb trithorax on certain cis-acting elements like the Polycomb response elements. We did not know the molecular mechanism of the function of Polycomb and trithorax. So the Vincenzo Peroda's lab, Vince Peroda and Jürg Muller's lab, so two labs, they were the ones, they published in Cell back-to-back papers. They showed that enhancer of zest, it actually, and now we are going to talk about EZ. Enhancer of zest, it methylates lysine 27 specifically. Okay, so which means the H3 lysine 27 trimethylation, it became a mark which is catalyzed by set domain containing EZ, enhancer of zest, and they also showed that if you mutate, if you do site-directed mutagenesis in the set domain of EZ, the lysine 27 mark is gone, which means, or if you mutate lysine 27 and incubate it in vitro with the enhancer of zest, you don't find, after mass spec analysis, you don't find methyl mark on these in vitro reaction products of histones, which means the enhancer of zest is specific enzyme or protein which catalyze lysine 27 methylation of histone H3. Since the chromodomain containing HP1 was shown to recognize trimethylated lysine 9 in H3, these labs showed that Polycomb, which also contains chromodomain and which is part of Polycomb group proteins, it recognized via its chromodomain the trimethylated lysine 27 state and contribute to Polycomb group mediated gene silencing during cell fate. Not long after, it was also discovered that trithorax, which also contains Z domain, it methylates lysine 4. Lysine 4 in histone H3, and these lysine 4 trimethylated state was due to Z domain in trithorax. So what became common, that Z domain, the amino acid regions in enhancers as trithorax group SUA3-9, it is responsible for catalyzing addition of methyl marks on amino acid residues, not the same but in different contexts. For example, trithorax is catalyzing lysine 4, trimethylation enhancer zest is catalyzing lysine 27, and SUA3-9 is lysine 9. So this really led to explosion of a high degree of activity in the field of chromatin and epigenetics, because there were many proteins which contain Z domain. For example, in trithorax you have ASH1, and we learned that ASH1 is involved in catalyzing, the Z domain of ASH1 is involved in catalyzing lysine 36 trimethylation of histone H3, and lysine 36 is involved in, is present in, lysine 36 trimethylation is present in gene body when RNA pol2 is going through the coding region. Lysine 4 trimethylation, lysine 27 trimethylation, lysine 9, they are present in the promoter region or in PRE regions, the cis-acting elements. But lysine 36 trimethylation was discovered to be in the gene body. And that's proposed that, when RNA pol2 transcribes genes, lysine 36 somehow acts as a memory mark for recently transcribed genes. Anyway, so in the epigenetic cell memory, what we have now in our hands, let's have Polycomb group, trithorax group. So you have in Polycomb group we have two main complexes, which is called PRC1. It comprises of, PRC1 comprises of 4 different proteins. For example, it contains Polycomb, chromodomain containing Polycomb, it contains posterior sex chromes, it contains D ring, and it contains polyhomoid. This is PRC1. And the other complex is called PRC2, Polycomb repression complex 1, Polycomb repression complex 2. And it contains enhancer of zest, extra sex chromes, and EZ, extra sex chrome, and I forgot the name of another protein there. There are other members as well, let's say X and Y. Now, the model is that EZ, it catalyzes lysine 27 trimethylation on genes which are to be maintained in the off state by Polycomb. And Polycomb, chromodomain containing Polycomb complex, the PRC1, it then recognizes the trimethylation of K27 and it maintains genes in the off state. There is also another component they recently discovered, not recently, quite a few years ago, that ring protein, it ubiquitinates in histone H2A. In the C term of H2A, it's lysine 118, I believe. So lysine 118, it carries monoubiquitination, ring 1. Okay? And together with lysine 27 methylation and ubiquitination, so PRC1 then maintains genes in the off state, and this complex, it blocks the RNA Pol2 from transcribing a gene. I try to draw things here, but before I move on, so antagonizing Polycomb group, we have the trithorax group. Now, remember I told you it's a seesaw kind of thing. We really don't know. Nobody knows what determines that, how the balance will shift. Whether, it will be the trithorax group which will win, or it will be Polycomb group which will win, which means winning means gene will be off or gene will be on. But these chromatin states or the histone modifications, these are known to be exclusive. If you have lysine 4-trimethylation by trithorax group, which is the trithorax, the set domain containing protein in the trithorax, if lysine 4 of histone H3 is trimethylated, you don't have lysine 27 methylation. And vice versa. If you have lysine 27 methylation by EZ, PRC2 complex, followed by PC, lysine in the presence of lysine 27 trimethylation, K4 methylation is absent. Okay? So, however, in case of trithorax, the complexes are diverse. This is a much more diverse complex than Polycomb group. So, in case of trithorax group, for example, we have histone modifying proteins. For example, trithorax, Ash1, etc. We have chromatin remodeling proteins. Chromatin remodeling proteins or complexes, they are the ones which utilize ATP, and they can slide. So if this is a nucleosome, and if this comes under the action of chromatin remodeling, these two nucleosomes, they can be slide. They could be moved apart, and this chromatin, the DNA, this can be accessible to transcription factors or transcription machinery. So chromatin remodeling proteins, they basically, which are part of trithorax group complex, they don't modify histones, but they may facilitate histone modifying proteins, because you need to, make these underlying DNA or histone proteins accessible. And then the trithorax group also contains proteins which are part of general transcription machinery. But we will stick to histone modifying and chromatin remodeling. For example, in case of chromatin remodeling proteins, you have Brahma complex. BRA, Brahma complex. These are, ATP dependent switch SNF kind of ATPases, which slide nucleosomes right or left, okay, to make the DNA accessible for transcription. Now, let's get back to where we started, and we started at epigenetic cell memory. Let's say here you have cell fate determination took place, and eventually after successive divisions you have differentiation of cells. And this we said is the epigenetic cell memory. Now Polycomb and trithorax, and I remind you again, we said we will focus on, in case of epigenetic cell memory, for UBX gene. So this was the haltear and this was the wing of fruit flies, which are established, their fates are established. This is going to make wings, this is going to make haltear, and we said UBX is off here and UBX is on here. Now, if we just see the UBX, so you have the PRE, which is called in trithorax complex, it's called the BXT PRE, and same UBX gene. So this is same UBX gene in the haltear cells, in the wing cells, as we again and again learning that each cell contains same set of genes, same chromosomes. Now here you will have PRC1, PRC2 action, which is going to have the N-terminal tail of lysine 27 is trimethylated, and chromodomain containing protein is bound, and UBX is in the off state. And here you will have these trithorax group proteins, and lysine 4 of H3 is trimethylated and UBX is on and cell fate is maintained. Now, it's not as simple, it's not as easy as I have drawn, because what molecular evidence has shown that the trithorax group proteins are also present here, and Polycomb groups are also present here, regardless of the gene is off or gene is on. So the big question in the field is, what determines whether gene is going to be switch on by trithorax despite the presence of Polycomb, or gene is going to be switched off or maintained in the off state despite the presence of trithorax group. We believe a lot of cell signaling is there, and that's a big black hole in the field of epigenetics. So the signaling to the chromatin is missing. We don't know what are real signals which are maintaining or which are signaling to either Polycomb or trithorax to maintain genes in the on or off state. However, what you should know that this is not just a fly specific phenomenon. We started our lecture with the humans, and I told you Polycomb trithorax, although the characteristic PRE sequences are missing in mammals, we really don't know, because the sequences, PRE sequences in flies, these are very well characterized sequences. You can take a PRE, put it next to a reporter system, it will switch off a gene. Okay? But mammalian sequences, I mean, there are recently 2 papers from Kingston lab, I believe, they came, which showed that PREs exist in mammals as well, but it's still under debate. However, what is important that both Polycomb and trithorax, they are evolutionarily conserved. They are performing exactly the same function what we discovered in flies. They are performing the same function in mammals as well, in our cells as well. So here, a very important concept I would like to mention is reversibility. We said that epigenetics is reversible. Imagine during our development, you need to switch on this specific gene. Okay? So we'll stick to lysine 27 trimethylation and its reversal. Let's say we have, this is, PRC1 specific silencing. Imagine we have now, this is gene X, which is off because lysine 27 is trimethylated by PRC1, PRC2 combination. And what you have, lysine 27 trimethylation, which means Polycomb is maintaining this particular gene in the off state. However, what they also discovered that there is a histone demethylase. Histone demethylases, they come in different flavors. Specific for lysine 27, specific for lysine 4, specific for lysine 36, lysine 79, etc. I'm sticking to lysine 27 because I want to explain you the whole phenomenon of epigenetic cell memory led by Polycomb trithorax. Because these modifications are not acting alone. They are acting, like I told you, H2A, ubiquitination, lysine 27, trimethylation. They are acting in a combinatorial manner. And there was a proposal about histone code as well. So it's a very complex phenomenon. But anyway, the histone demethylase, which is called, in this case, is called UTX, it demethylases. So H3K27 trimethylation is gone. It removes this mark, and then since lysine 27 is now without modification, it is replaced by mark called acetylation. It becomes now H3K27 acetyl. And acetylation is what? Gene activation. Okay? The serine, which is lying next to lysine 27, which is serine 28, the phosphorylation of serine is known to play a role here in reversal of lysine 27 trimethylation. And the lysine 27 becomes acetylated by a protein called Krebs binding protein, CBP, which is part of trithorax group complex. And once you have reversal, it is followed by lysine 4 trimethylation, so H3K4 trimethylation by trithorax. So look, CBP is also part of the complex in which trithorax is present. So they are reversing K27 trimethylation and bringing in now exclusive lysine 4 trimethylation, and gene is going to be switched on now. Here, gene from off will become on because the epigenetic mark is reversed like this. So this intrinsic and a very complex action between Polycomb and trithorax, they are involved in maintaining the epigenetic cell memory of the cell fates which are established early during development. And that was discovered, that was very well characterized in flies. The conservation of Polycomb and trithorax, not only in terms of Hox gene regulation, because homeotic genes, bithorax complex and Danapedia complex, they are fully conserved in mammals, in us. There are 4 clusters, Hox A, Hox B, Hox T, etc. And the organization of Hox genes is 100% similar to fly genes. Even their expression pattern from anterior to posterior is exactly same as flies. And we call this collinearity because their organization on the chromosomes as well as their expression, spatio-temporal expression in mammals is also fully conserved. And their expression, active or silent, is also maintained by Polycomb and trithorax. Last 20 years I have discovered that Polycomb and trithorax are not just maintaining Hox genes. PREs were discovered beyond Hox genes. PREs were discovered in nearly 300 different genes in flies. And to add new twist or complexity to the story, it was discovered that Polycomb group, the H3K27 trimethylation in mammals, it actually controls 5-methylcytosine. You remember when we learned epigenetics, we talked about DNA methyl mark, DNA methylation in CG context, which is maintained by DNA maintenance methyltransferase. This is controlled by lysine 27 trimethylation. And also in terms of constitutive heterochromatin, where lysine 9 methylation, SUA3-9, but in mammals, I'm talking about SOV-H1 and SOV39H1 and SOV39H2, they regulate DNA methylation in constitutive heterochromatin. So it's not just either or, because in flies there's very little rudimentary DNA methylation in first 30 minutes. So when it comes to DNA methylation, we talk in greater detail in mammals, in plants, or in case of animals. I would also like to mention in Arabidopsis a reverse relationship was discovered. In Arabidopsis it was discovered that CpG methylation, the cytosine methylation in this context, this is the one which is controlling lysine 9 trimethylation in heterochromatin. So these covalent modifications of heterochromatin, they interact with each other. There's a crosstalk, and they maintain these reversible or these epigenetic states. So we are going to stop here for covalent modifications related to Polycomb and trithorax. And in the next part, we are going to just talk about DNA methylation just briefly. That will be the last part of this epigenetic cell memory, because we will talk the transgenerational. We'll talk about, inheritance of epigenetic information from parents to offsprings in relation to DNA methylation. So stay tuned.